Good afternoon. Thank you for joining us, and welcome to the first episode of our Lab Life Simplified webinar series, Stem Cell Culture Shock No More. Before we start, allow me to remind you about the ground rules for this webinar. Please turn off your cameras during the webinar and keep your microphone muted throughout the session. Questions can be asked through the QR code for Canva Live. You can ask questions in both English and Thai. If you are unable to scan the QR code above, please go to canva.live and type in the code displayed. And these are our Facebook and our LinkedIn. Facebook is for Thai people and LinkedIn is for global. You can scan the QR code to follow us. If you are aiming to elevate your research and keep up to date with the bite-sized lab tips and industry updates, please kindly follow our Facebook page and our LinkedIn page. My name is Lucas from Atlantis Bioscience, and I will be your host for this session. Just to tell you about Atlantis Bioscience, we are a translational solutions provider providing services from bench to bed in Southeast Asia and beyond. We were established in 2016 and represent a variety of brands, as displayed here on the right. Beyond lab equipments, we also offer workflow and translational solutions for biomedical and healthcare research. We specialize in curating and developing solutions in 4 main focus areas: cellular and molecular study, drug development and delivery, cell and gene therapy, and bioprocess and alternative protein. Today, we are with Dr. Rattajak Rangsiviwat, or Dr. On, from the Department of Anatomy, Faculty of Medicine of Srinakhrin Virod University. Dr. On has extensive research experience in stem cell research in both humans and animals. Today's session will help you reskill your stem cell techniques with tips from a veteran in the field, while also helping you to troubleshoot any problems or shortcomings you come across in your research. For our agenda, we will first give an introduction to stem cells, followed by how to set up a stem cell lab. Then we will explore tips for successful stem cell culture and its applications. We will then wrap up with a Q&A. Now I'll hand it over to Dr. On to share his experience.
So good afternoon, everybody. Could you hear me? Okay. Thank you so much for Atlantis Bioscience for giving me this opportunity for sharing you guys about stem cells. But first of all, I would like to say that this talk is not a really in-depth in scientific talk. Just want to share about how things work in the laboratories. Of course, I know that after I search for the audience, I know there are many people in different backgrounds. So I will try to make everything simple. And I know that some of the audience are not scientists, but from the company. So we can share the idea and experience. How can we run the stem cell laboratories? So as I mentioned earlier, so today my talk will be a bit simple, but the simple thing like star egg or omelette can make things different, can change our success, or it can bring us to the failure. But however, so let me start a little bit about the biology of the stem cell before we going up how it works in the laboratory. So there are 3 important 3 characteristics of stem cells. So the major thing is that the stem cell, they can renew themselves. Renew means they can replicate themselves through the cell division. And also the stem cell, they are unspecialized cells, meaning that they do not have the ability to perform a tissue-specific function. So it means like they are young. So when they're young, they can be controlled. They can change to anything that you want. So it means that they can be induced to become any cells in special functions, like neural stem cell can be induced into the neuron or the supporting cells, or the stem cells can be induced into the beating cells of the heart. So let me explain a little bit about the biology of the stem cell by using the pictures. Okay, so in this picture on the left side, you can see the group of the cells. They are orange color. So once when they divide symmetrically, symmetrically. Remember, if they divide symmetrically, so they can give rise to the daughter cells with size, with similarity of the size, genetic background, and everything have to be similar as a mother cell. But once take a look on the right side, if they divided themselves asymmetrically, so they can change to be a specific cell, either a muscle cell, nerve cell, blood cells, or any cells. But once again, after division, they still remain their stem cell characteristic in order to can renew themselves for another of time. So just summarize once again, before we move to another session, the stem cell that can renew themselves for a long period of time. Take a look of the word long period of time or long period of cell division, because this word can confuse the researcher, or you guys, you might think that the stem cell can be grow in a laboratory forever. No, they are not. In some condition that's not certified the cell to grow, they might die. Okay, but again, stem cell can be unspecialized and it can be induced to become the cells with a special function. Sorry. And then when we know about the characteristic of stem cell, but how they come from, where can we find them? Are they in our body or they are somewhere? So let's take a look where we could get the stem cell. First of all, there are different kinds of the source of the stem cells. The first thing, we can get the stem cell from the embryo, human embryo. A group of specific cell called inner cell mass. The inner cell mass can be give rise to our body or animal's body. So it means the group of those cells, they can change, they can differentiate into a specific cell. But this group of cells, we have to collect the inner cell mass stem cell from the blastocyst embryo from the human. It means if we want to get these cells, we have to destroy the life. We have to destroy the embryo. So fortunately, the scientists discovered that we can change the skin cell, for example, or somatic cells by using a reprogramming factors. Let's say like we manipulate the gene of those somatic cells, then they return to be a specific stem cell we call induced pluripotent stem cell, which have a similarity of phenotype and molecular level similar to those embryo derived from the embryo. But today I'm not going to focus on these two kinds of cells. I will focus more with the stem cells that derive from our organ or the body in our body. So mostly they are known as a mesenchymal stem cells. In this picture, you can see that almost every organ in our body contains the stem cells, such as bone marrow. If we collect the cells from the bone marrow, we could grow them, and we could have them a mesenchymal stem cell, a hematopoietic stem cells together with mesenchymal stem cell from the bone marrow. Other tissue like adipose tissue, muscle, placenta, or umbilical cord, even dental tissue and skin, they all have the stem cell reside in that area, in that specific area of that tissue. So once when we extract the stem cell from those tissue, we can grow them in the laboratories. And this block below is the potential application of the mesenchymal stem cell, such as we can use the stem cell for bone and cartilage regeneration, using for treatment of cardiovascular disease or wound healing. But please keep in mind, this is just a potential application, a potential. It doesn't mean this is a standard treatment. So we have to think about if we use those stem cells for treatment in specific disease, so it's just a potential. It can have some risk as well. And this slide will be the last one for general biology. So when we think about we want to grow the stem cells, we have to think about the purpose of our study or our research or our application. If we use the stem cell for culture or differentiation, we have to think about how we can control them. Or we are growing the stem cells with different methods like addition method or suspension method. It really depends on the kind of stem cell. For example, if you get a stem cell from water jelly, so whether you use them to grow them on the platter dish, but if you culture the NK cell or hematopoietic stem cell, better to grow them in suspension. So this is very important to understand how their behavior, how they like to grow in the laboratories or in the incubator. But also the morphology of the cells is also important for us. Why it's important? Because if we don't know their morphology, we don't know what happened in the cell. We have to know, like if you are falling in love with somebody, you will have to know the behavior, you have to know their face or the look of those person. Then you can, I don't say manipulate, but you can know how to deal with that person. Same as the cell. If you know the morphology of the cell, you will know how to manage the cell. All right. So I hope that a very short introduction about stem cell biology will help us now, all of us in this meeting today to go further for the example. And I want to say it's not really a tip, but it just experience an example that we experience in our laboratories. So today about this session, I will explain to you as a scenario. In each scenario, you will see and you can exchange the idea. In the case of your laboratory, what did you do? What you manage with that scenario? But in our laboratories, so we will tell you what we have done and what we are going to do. So let's start with the first scenario. I think many people of you have been assigned by your PI, your principal investigator, or your lab leader to set up the stem cell laboratories. Many years ago, I got assignment from my PI to set up a stem cell laboratories. So there are so many things that you have to think about. What kind of laboratories that you're going to have? You're going to have a GLP laboratories, or your company going to set up the GMP facility. So you have to follow the regulation. You have to follow the regulation, but also the more important thing, you have to follow the biosafety level of the cells that you're going to grow, the kind of the laboratory that you're going to set up. And that things I'm not going to talk about today because maybe in the future, Atlantis Bioscience will organize this kind of topic. But what I would like to emphasize all of you today, so please remember that the cells that we are using, it can have a risk because we get the sample from the donor. Even though we make a screen test for the donors, they are free from the disease, but anything can happen because it's biological sample. And in this picture, I show you that what we have seen. Zero pipette or pipette tips is considered as a sharp material. The way we have to get rid of them, we have to follow biosafety level 2, level 2, or anything that related to. Because of this, I experienced twice. In the past, I worked in the hospital where they have a system of destroying of management of the waste, biological waste. So we have to put in the red bag. But then my student, we have a new trainee coming, and I was not in the laboratories, so we put the zero pipette in the plastic bag like this, and you can see on the picture above on the right side that the zero pipette protrude from plastic bag. So this is very important. So you have to keep in mind these things. You have to keep in the a box or some container that reduce the risk of accident. Not only the way to manage the work in the laboratories, the workflow in the laboratories, the biological waste that you have to manage, but more important things is a note. If you would like to meet the criteria of GLP, GMP, you have to take a note. You have to make a record of everything, the batch of the medium you use, the person who do the experiment, or even the result during the day. Okay. Remember, work without taking a note, it means no work. And I show you, this is my lab book. when I work in Japan. So I have to write it down, take the pictures, and every week I have to send this a lab book for my PI to check. So this is very important thing because I think now in Thailand, many laboratories don't do this. And especially if you're working in a private bank or private laboratory, this is very, very important. So please take a note and keep everything, because if something wrong today, so we have get back and check what happened in the past. And the scenario two, when we set up the lab, so because we don't have, I mean, in my lab, it's not GLP, it's not GMP, it's just a clean lab. And so we don't have enough money. So we bought the centrifuge. We bought a centrifuge, and even myself and my student asked which type of centrifuge we should buy. We should use a fixed rotor or swing out rotor. So in the case, I think it can work both way, but mostly when I use the swing out rotor. So in the pictures on the top panel, you can see when the centrifuge works, how the tube going to float, either like in the swing out bucket or in the angle, I mean fixed angle. But the most important thing is that when it stops, when the centrifuge stops, it had to brake to brake slowly. It's not immediately brake. Otherwise, the cell pellet will float up in the suspension. So this problem, we had it before. And the problem is that we book a very cheap centrifuge, like 20,000 baht. So it cannot control about the brake. So now we have a better one. So if you think sometime the brake down of the centrifuge cycle doesn't work properly, change it, fix it, because otherwise you will lose the cells in the suspension. Some of the cells may not be sedimented. Okay, so there are already 2 scenarios. We have 7 scenarios before we start the Q&A session. The scenario 3, when we start the stem cell laboratories, I'm not sure if some of you have this question in your mind. I think you have it, right? You grow the mesenchymal stem cells. They are different basal media in the market. You take a check on the literatures. Some say DMEM, some say DMEM low glucose, some say alpha mem. Actually, I think the basic, because they have a different type of basal medium, they have we have to consider about the concentration of the glucose, because glucose is a major source of energy for glycolysis for the cells to take for that to grow. And also for glutamine, it's also have to be supplemented in the medium. But however, you can prepare your own medium by combining the basal medium with other supplementation, because it gives you a flexibilities and you can control the cost of the research. But however, like what I said earlier, different cell type might need different composition, different supplementation. So you have to take a look, you have to make a list of what really the cell needs. And once again, even the same cell line, same cell type, during the routine cultivation, they might need one basal medium. But once when you would like to differentiate them into a different cell type, you will need to change to another basal medium. So in this case, you have to study and take a note. And of course, you have to make a check from the literatures and other laboratories. But once again, even in the literature, have the procedure, have the a procedure written, but in your own lab, you have to set up everything and find out which one is the best procedure in your lab. In these pictures, I show you about the morphologies of the adipose-derived stem cells from a goat. They use 10% of fetal blue white serum, but different basal medium. You can see A, B, and C, A, B, C, and D. The morphologies are different. Especially take a look on the panel D doc. So you see the cells are flattened, they don't grow nicely, so they start to die or differentiate. But in my case, I usually use MEM alpha mem to grow a mesenchymal stem cell. So the morphologies always look like panel A. But this picture just want to show you that just the basal media are different. The serum the same, the percentage of V-double-Y serum are the same. And that come to another questions, another scenario to see that do we need to use the serum, or we need to use this, a media without the serum, or we call serum-free media, to grow our stem cells? The answer is it really depends on your work. If you are working in the normal science, I mean, in the basic research, probably you still have to need to use the serum. But if you would like to use the experiment for clinical trial, I would suggest better use serum-free, because serum can affect a lot of things, a lot of morphology, molecular biologies of the cells. And of course, if we use the stem cell for clinical trial, the serum contain of the cells. The serum might change some epitope of the cell that might create some allergy or can affect the reaction of the patient or the animals in the clinical trial. So this is really important. You have to think. But for example, if you start your work with the serum, but when you would like to change to the serum-free or animal free product, I would suggest not to change it immediately. I mean, for this passage, you grow them in the serum, but in the next passage, you can reduce the serum a little bit and you add more serum-free condition until the cell can adjust themselves. And then you can see how things go and how it works. I think in this case, it's not gonna make the cell shock like what we say, no shock for cell as well. So this is the part that I show you in, I mean, the result that I show you I have done in the past. We grow the stem cells from skin, human skin, in the V-double-Y serum in comparison with the serum that we derive from cord blood serum. So we could see that the cells that grow in the cord blood serum attach to the plastic dish, to the surface of plastic dish, slower, much slower than the V-double-Y serum. But 24 hours after we grow them, so they attach nicely, not many floating cells. And after culture for another 3 to 4 days, so they can multiply nicely. And our results showing that the human skin fibroblast is grow much better in cord blood serum. And our idea is try to find a good serum for stem cell that we grow in the laboratories. Our idea is that we would like to use the serum that derive from the same species of the cells. I mean, in the past, in a former slide, I show you to grow the human stem cells using the human serum. In this one, I try to change the serum from V-double-Y serum to a goat serum in order to grow a goat muscle stem cells. So you can see on the left side, so they are very low density, lower, let's say, a lower density than on the right side, which is the goat's stem cell growing at 10% goat serum. And this is another experiment that we have done. We grow the human stem cells derived from the endometrium tissue, a human endometrium tissue. So we try to find which serum is better for them to grow. So we compare with the V-double-Y serum, which is almost every laboratory use. And in the literature, they use 10% V-double-Y serum for growing stem cell from endometrium. But we compare with the human serum, but with two bands. And we found that the stem cells of endometrium on the cell that growing in human serum is better than V-double-Y serum. Okay, so we go to the next scenario about isolation of the stem cell from a tissue. many people isolate the stem cells from fat tissue, for example, or water jelly, cord blood tissue. So in the laboratory, you use the enzyme to digest the extracellular matrix. Then you can get the stem cells from the tissue. But my student came to me and asked, I follow the published protocol, but why I get the heterogeneous cell population, but not only mesenchymal stem cells. Sometimes they could get some cells look like epithelial cells. And this picture just to show you that this is a reality. You can see on the corner, on the right corner, you see some pictures that look more fibroblast-like shape. And on the right side, the cell are smaller and packed, more density than on the right side. So this is a sample that you might get this kind of mix of population of the cell during the isolation by using the enzymatic digestion. So in our case, we also using the tissue explant in order to isolate the stem cell from the adipose tissue and watan jelly, because we think they might help to reduce the heterogeneous population of the cell during the isolation period. Then when we get this kind of cell, a mixed population, what we do next? Of course, if we have money, we can use cell sorting technique. To make it simple, we can use what we have done. We use a magnetic cell sorter, but again, you have to use antibodies to attach to the surface of the cells and then let them pass through the magnetic sorter. So then you can get and wash out the positive cell. So in this case, on the right side, you can see that the population of the cells are more homogeneous. So in this technique, we could get a better result of homogeneity of the cells using the enzymatic dissociation. But in different type of stem cells, like hematopoietic stem cell or NK cell, you might need another machine with more advanced to isolate the cell, because they are small population. You don't need to lose them. So maybe better that you invest in the better equipment to isolate them from the starting material, starting tissue. And so that was the fourth scenario about isolation of cells already. Now we get the more purified stem cells. But once when we grow the cells, especially in the laboratories that you grow many different cell type, and you might face some problem that there are other cell type that growing in the stem cell culture dish. That means today you're growing one cell type, but the next day or 2 days later, you check, there are some other things, some other cell type growing in the same plate. I let you play it a little bit on these pictures. Can you figure out, can you find out? There are two different morphologies of the cells in the same petri dish. I'm sorry if I think I cannot point by the pointer here in the pictures, but you can see, yeah, thank you so much, Lucas. Yeah, left a little bit left. Yes, correct it. No. Yes, that's it. So that was the first group. And then on the left side of this globular cell, you see the different morphology. So different morphologies mean, the first things mean they are different cell type. Yes, indeed, they are different cell type. The narrow one, the thinner one are the mammary gland carcinomas. And the rounder one, they are lung cancer, lung carcinoma. They are different cell type, but it mixed up together. And the next example, which is even strange. So there are two different cell type. If somebody who work with iPS cell, ES cell, they might think this, wow, this is a good colonies of iPS or ESL. Actually, no, this is a cancer cells, but they accidentally jump. I don't say jump, but accident by researchers, I think by myself as well. They mix with the mesenchymal stem cells. How come it happens? If you work in the same laminar flow, well-safety cabinet, and some people would like to make different kind of cell type together because of want to save the time. So they might use the same pipette or zero pipette. In this case, I talked with my student, it was very, very interesting. She used zero pipette to remove the culture medium and remove the medium and throw it out, and then use the same zero pipette to remove another dish of the medium. So I mean, the mix of the cells are really because of the zero pipette that stick, the floating cells that are in the culture dish that stick in the zero pipette and then move to another dish, and that's why we could see this kind of contamination. This is very important. I said I use this scenario in this talk because of if you work with two different cell type, it's easy for you to figure out. But if you work with only one cell type, for example, if in your lab, you have a mesenchymal stem cell derived from adipose tissue, but different donors, or if you use you work with Watan jelly stem cells, but you work with different donors, but you would like to save your time by taking the cell from the donor one together with the donor number two, and they use the same equipment that might contaminate with two donors, but different cell type, you cannot notice. That means it might mix up, that prejudice might mix up the genetics of two donors together. And if you use it for clinical trial, they might give you a surprise result. And we go to the next scenario. When you growing the cells, so sometimes your cells growing very slow and look unhealthy, even though in the previous passage they were very nice, they grow very nice shape of fibroblast-like shape. But once this time the cell look unhealthy. In this picture, I'm sorry again, there are some cells that unhealthy. You can see some fragment in the cytoplasm, and they start to not have a fibroblast-like shape, not elongate fibroblast-like shape anymore. They have like epithelial cell, they get a bigger shape. So it doesn't mean that they don't want to grow anymore. So despite of they are getting old or getting aging, they are a very complicated contaminate in the laboratories, that is mycoplasma contamination. So I will really like to convince you to check the mycoplasma contamination in the cell culture in the lab, because they can ruin all the work. Sorry. It can ruin all of your works, and it can create a bad result. Of course, you can protect it by having a good hygienic work, I mean sterile work, but it really necessary to make a check, almost regularly check, like what we have done in the lab. In our lab, we check by using a colorimetric method, like you see on the left side. We check just to see the color, but this is just a pre-screening. If we would like to be sure to make sure that we have a clean cell, so we have to send it to the third party to make a check for a mycoplasma check. And the last scenario that I would like to share to you, it's, I think most of us doesn't want to work on the weekend, right? Me neither. But by chance, not by chance, even myself, I've done it when I was a student, but also my student as well now. We try to see the cells on Thursday or Friday without checking. So we just say, okay, passage with a low density. On the left side, you can see this is the result of the low density of passaging the cells. And on the right side is a high density. So you can see a lot of cells in the petri dish. So in this case, yes, you save your weekend, but you don't save the cells. Growing the cells to low density, they cannot grow nicely because they need a cytokine, they need communication. But on the right side, if they are too packed, they are really like in the slum, so they're not grow well. They will change their biology and morphologies of the cells, and they can create another trouble. Yes, simple thing to do is to count the cells. And you check the growth rate of the cell. What is doubling time of the cells? If you start to count the cells all the time and you have the doubling time of each cell line or each donors, you can manage your life. You can manage your weekend. You can manage that, okay, on which day they are going to reach a plateau and they will stop dividing. So that day you have to do something. In this case, I really convince you to count the cell almost every time and make a summary, make a record of each cell line. Because nowadays we don't need to use a manual work like what we do, a lot of what I show you on the left side. You can use an automatic machine to count the cells. That's easier. And at the end of my talk, I would like to end up here what we are going to do, our ongoing research. Mostly we use the knowledge and the technique of stem cell biology to do in vitro culture meat. In vitro culture meat on the left side, you can see the muscle fiber, muscle tube that occur after differentiations. And also we use the stem cell from the cat for preclinical trial by using the stem cell for wound healing. And this cat now, the wound is healed and we cannot use a surgical reconstruction of the skin. So we have to use the stem cells injection. And also we are doing the organoid culture by using human endometrium, and we do a modeling of human neurulation. The development of neural system of the human by using the human embryonic stem cells. And other two things that we are working now is using a molecular technique to analyze the pre-implantation human embryo before we transfer the human embryo into the mother. So we do a genetic test before that. And also the last one we are doing now is using the stem cell as a factory to produce the extracellular vesicle for research and preclinical trial. And thank you for your attention. Before I finish, I really like this quote, and I keep in my mind all the time to be like stem cells. Not because I'm crazy with stem cell, but because I would like to differentiate myself. I would like to differentiate myself from the others. So that's why I keep working with the stem cells. Okay, thank you so much for your attention, and I'm happy to take questions.
So one of the question is, what are the most critical best practice for maintaining stem cell viability during the culture?
The most critical.
Yeah, best practice for maintaining cell viability.
Okay, I said it in general. It's not only about I don't mean research or clinical trial or stem cell bank, but I would like to say that regular check of the phenotype and other cells. Like if you grow a mesenchymal stem cell, you better to check the characteristic of the cells before or after cryopreservation, and also the contamination. You have to check it. That's the best practice. Viability.
Yeah. Okay, the next question is, can you share what is the best technique for passaging stem cells to maintain their pluripotency?
Okay, in this case, I think maybe you mean the iPS cells or embryonic stem cells, I guess. So I would like to, there are two things, mechanical passaging or enzymatic passaging. And enzymatic passaging, because it can create small clumps, so you might have to use a ROCK inhibitor to help them not to go apoptosis. But keep in your mind that using ROCK inhibitor too much, they might change some morphologies. And in our case, it even induce into neural differentiation by using ROCK inhibitor. So I would suggest that sometimes you use a mechanical isolation just to help, like cutting. You may use some equipment that cut the colonies into small size before using the enzyme. In this case, you can reduce the timing of the enzyme before passaging.
โอเค อันนี้ภาษาไทย อาจารย์ครับ ถ้าอยากขยายแอปพลิเคชันเนี่ย ควรระวังอะไรบ้างในการเพาะเลี้ยงเซลล์แบบ
โอเคคำถามของแอปพลิเคชันเนาะโทษครับคำว่าแอปพลิเคชันต้องดูsorry okay I answer in Thai first ถ้าจะขยายแอปพลิเคชันขึ้นอยู่กับว่าเราจะใช้ไปเพื่ออะไรครับก็ถ้าหากว่าจะใช้เซลล์ จริงๆระหว่างเอาสเต็มเซลล์ไปใช้เลยกับเอาสเต็มเซลล์ไปผลิตเอ็กโซโซมแน่นอนครับมันจะต้องตัวคุณภาพของเซลล์ต้องดีแน่นอนเซลล์ต้องเฮลตี้ครับต้องไม่ให้เซลล์เข้าเอจจิ้งไวนะครับที่สำคัญก็คือคอนโทรลของคอนโทรลเซลล์เดนซิตี้ในระหว่างการเรียงให้ดีครับokay so my answer is that in order to help the cells for application how can they focusing during the culture system culture the cells I would suggest you to control the cell density I mean you have to check in which density that they grow nicely they don't grow aging fast so then follow that density but keep in your mind the longer I mean the higher passage number the cell will change their behavior and don't use the old I mean cells high passage number of the cells it can give a bad result
Okay, so just to translate the questions for those who don't understand Thai, the question was, what considerations should be made when scaling up stem cell cultures for larger applications?
okay oh.
Do the serum used for stem cell culture need heat inactivation?
I thank you so much for this question because I have this idea before, but I forgot to explain during my presentation. Actually, I use both inactivation and non-inactivation because inactivation will stop the activity of complement. But in my case, I try to use both inactivate and non-inactivate to grow to the same cells, same cell line. One cell line doesn't like inactivate at all, don't grow at all, but they like non-inactivate. But some cell line they like inactivate. So really, it's really complicated. But from literature, they say some other companies say you don't need to do it.
Is it necessary to use the same batch of serum to get a more reproducible result for stem cell culture?
Theoretically, yes. Theoretically, yes. But once again, you take a note of that batch. But if you think you can get the same batch, do it. But if you cannot get the same batch, you have to try to make a test when you receive a new batch first. But in this case, the same batch is very important for in my experience, very important for cardiomyocyte differentiation because different batch give a different result.
Okay, do the short-term tandem repeat STR analysis, is it needed for stem cell culture?
Sorry. It needs in case of you would like to check the source of the cells. In general, I mean, in the research, we don't do the short tandem repeat analysis, but in some case, so they do it.
Okay, what are some common mistakes you see new scientists make in stem cell culture and how can it be avoided?
Changing the culture medium. First thing I have seen, like what I explained, okay, in the lab, they said, we you change the medium every 3 days. They fix it every 3 days. But what I want to say, take a look at the cell first. Some cells like to be changed. The medium should change every couple of days. But in our lab, sometimes we change the medium, not 100%, we change only half, but more often. Like instead of you change the medium every 3 days, I change medium every every other days, but only 50% remove and add a new one, 50%. In this case, you can keep some parakine or cytokine that stay in the culture dish, but you adjust the pH and remove some lactic acid out from the culture dish that might help the cell. This is the mistake. And other thing is that common mistake is don't take a note. I've seen a lot, really. If you have a new scientist who would like to get a job at your company, check it out. If that person really like to take a note, take it. Otherwise, it will ruin the work in the lab.
Okay, just to add on to what Dr. On is saying, routine mycoplasma retention is also one of the more important practices in keeping
Yes.
your stem cells healthy, right?
Yes, yes,
So
of course.
our company Atlantis offers both micro-decon spray as a preventative measure, and we also have a mycoplasma detection kit using PCR or qPCR method for your routine screening. So you can approach us through the channels that we mentioned before to know more if you're interested. Right, now let's move on to the next question. Publications showed the usage of both human serum and human platelet lysate in MSC culture. How would we know which one is to be used for your culture?
I wish, okay, let's say human serum is a mixture. They are undefined material inside the serum, because they can have a hormone, they can have or other stuff, but hip pellet lysate is only from pellet. So it can create some growth factors that necessary for the cell to grow. Let's say, sorry, pellet lysate is more defined than human serum. So if you have a better money, if you have more money, so you can go to pellet lysate. You can control, you can know exactly what happened with your cell because of the serum. Like what I said, they can get from different different donors, and they can have more hormones inside. So different batch will create a lot of variation. Sorry. Yep.
Right, let's move on to the next question. And we think that's all.
Okay.
Right. There is one latest one though. Yeah, is it necessary to use inactivated serum to culture iPSCs?
Okay. I would suggest not to culture iPSC with serum, like what I said, like what I answered in the previous question, because serum contain unknown factors that we don't really know what gonna create the effect. It will affect iPS cells. So no need to answer about inactivate or not inactivate. Better not to use the serum for normal culture, but we use the serum-free or defined media. There are many companies selling the defined media for growing iPSC. But once when you would like to differentiate iPSC into different cell type, some protocols still need to use stem cells. Still need to use a serum for differentiation. You know, if you follow that protocol of using the serum for differentiation, not for culture, for differentiation, better to use inactivated.
All right, and I think that's all the questions. So thank you, Dr. On, for sharing your valuable experience for people who want to start stem cell culture, or tips for people who are already doing stem cell culture. If there are any parts that you missed or would like to listen again, don't worry, because we will send you a recording of this webinar shortly. If you are still hungry for more tips and tricks for stem cell culture, feel free to visit our website, blogs, for more comprehensive information. And if you are interested in some products that we have, such as stem gold for our stem cell culture, you can contact us through these channels as well. Thank you for joining us in our first episode, and we hope to see you again in the future. Have a great day, everyone.
Thank you so much. สวัสดีครับ